IB Biology Cell Respiration, Part 1, introduces some foundational ideas for understanding cellular respiration at the SL and HL levels. This movie also includes material on oxidation reduction reactions, which is appropriate for HL students only. For SL students, you will be accountable for material in Cell Respiration Part 1 and Part 2 as indicated by the syllabus statements embedded in each movie. Be patient with the mixture of SL and HL concepts. The IB syllabus does not do a good job of cleanly dividing cell respiration into SL and HL sections. The essential ideas are: cell respiration supplies energy for the functions of life, and secondly, energy is converted to a usable form in cell respiration. Here is the outline of all the movies available for the cell respiration unit. Use this outline to find the movie you need for review. This movie is focused here. Cell respiration is central to the metabolism of a cell. This diagram displays hundreds of metabolic pathways that occur in a cell, and this is just a fraction of them all. You can see the outline of the mitochondrion here. The role of the mitochondrion as an organelle that transforms energy is vital to life. Here are the first two IB syllabus statements. Define cell respiration. Cell respiration is the controlled release of energy from glucose or other organic compounds to form ATP. Secondly, state that ATP from cell respiration is immediately available as a source of energy in the cell. ATP is used for cellular energy needs: active transport, synthesis of large molecules, motion, which involves sliding protein filaments, and cell division. You should remember that the mitochondrion, seen here, is an organelle centrally involved in energy transformation. You will read that the mitochondrion is the cell's powerhouse. As IB students, we need to do better than this. We need to understand the mitochondrion in greater detail. Here is the general equation for cell respiration. Glucose and oxygen are the principal inputs, the reactants. Carbon dioxide and water are the outputs, the products. Study this equation. You should be able to balance it if asked. This diagram provides a big picture view of energy transformation in cells, starting with the absorption of solar energy by plant cells. Chloroplasts, the organelles within plant cells, absorb solar energy along with carbon dioxide and water to manufacture organic molecules, such as glucose. Organic molecules are then broken down by the mitochondrion to produce ATP that is used by the cell to do cell work. For example, active transport, movement, and the building or the synthesis of large molecules. As an animal, you don't synthesize organic molecules from photosynthesis, but you ingest large organic molecules in the food you eat. Then your cells break down the organic molecules in the mitochondrion to produce the ATP for your cell needs. This diagram broadly displays energy transformation in a cell. Organic molecules such as glucose arrives to the cell either by photosynthesis in a plant cell or ingestion in an animal cell. The energy contained in the large organic molecules such as glucose is released and ATP is synthesized. The synthesis of ATP is an endergonic reaction. The energy from glucose is released to produce ATP. Then ATP is broken down in an exergonic reaction that releases energy available to perform cell work. The remaining products from the breakdown of ATP, which are ADP and an inorganic phosphate known as PI, are then put back together by cell respiration in the endergonic reaction. The endergonic reaction requires an input of energy to put the ATP back together, and that comes from the organic molecules available to the cell in the food that you eat or in the glucose that's produced through photosynthesis in a plant cell. Here is a micrograph of a mitochondrion, and here is the general equation for cell respiration. You need to memorize this equation. Cell respiration has an input of an organic molecule, glucose, as well as an input of oxygen. Carbon dioxide and water are the products to be released as waste from the cell. Glucose is a molecule with substantial available energy in its bonds, energy that when released is used to synthesize ATP from ADP and PI. Structurally, the mitochondrion has an outer membrane and a second inner membrane that is quite folded. The interior solution is called the matrix. The inner membrane is the region where most ATP is synthesized. The inner membrane of the mitochondrion has a huge quantity of protein embedded in it as compared to other membranes within a cell. This is evidence of its large role in the production of ATP. As I've mentioned, cell respiration is an exergonic reaction, an energy-releasing reaction that drives the endergonic reaction of ATP synthesis. The energy released when glucose is broken is used to synthesize ATP. The breakdown of glucose is catabolic. Large molecules like glucose to small molecules like carbon dioxide and water. The energy from the glucose breakdown is used to drive the anabolic synthesis of ATP. Small molecules, ADP and P, to large molecules like ATP. This is anabolism, energy storage. Cell respiration is the controlled release of energy from glucose or other organic compounds to form ATP. You can see the reaction here. Glucose, a relatively large organic molecule, is the reactant in cell respiration. Glucose is broken down into carbon dioxide and water, the products. The progress of the reaction is shown here along the x-axis. Glucose as the reactant has substantial energy. The reaction occurs and the products have less energy. The released energy, the difference between these two, is used to drive ATP synthesis. The energy in glucose comes from photosynthesis, where chloroplasts absorb solar energy and transform that to the chemical energy of glucose. Cell respiration, an exergonic, energy-releasing reaction, is seen as an equation here. The controlled release of energy from glucose drives ATP synthesis, and this is an endergonic reaction. In a cell, ATP is hydrolyzed, broken down, to do cell work, active transport or motion, or the synthesis of large molecules in the cell, like protein synthesis. The products of ATP hydrolysis, which are ADP and PI, are put back together again by respiration, energy coming from glucose breakdown. Here is a diagram to help you locate all of this within a cell. Cell respiration happens mostly in the mitochondrion. Respiration is the controlled release of energy from glucose to drive ATP synthesis. ATP leaves the mitochondrion for the cytoplasm, where it is broken down to do cell work. ADP and PI return to the mitochondrion to be put back together again using the energy from glucose breakdown. Here is the relevant IB syllabus statement. Cellular respiration serves to provide the cell with ATP. ATP is immediately available as a source of energy to the cell. Carefully describe the structure of ATP and explain why its structure makes this molecule such a good energy carrier. ATP is adenosine triphosphate. ATP is a nucleic acid relative. Here is adenine, which is one of the 4 bases of DNA and RNA. Adenine is covalently bonded to ribose, the same pentagonal sugar in RNA, and then 3 phosphate groups that dangle from the number 5 carbon of ribose, Adenosine Tri-Phosphate. When ATP is hydrolyzed, broken down, a phosphate group breaks off, releasing energy, the energy to do cell work. ATP is a good energy molecule, not so much because it holds a lot of energy, it really doesn't. But ATP is a good energy molecule because the bonds between the phosphate groups are easily broken due to the negative charges that repel one another, facilitating the breaking of bonds. Here is the next IB syllabus statement, and it's for HL students only. Oxidation reduction detail helps lay some groundwork for moving forward. Some of what I'm about to present should feel familiar from chemistry that you've studied previously. Here's the statement. Define the terms oxidation and reduction. Explain the processes of oxidation and reduction. State that cell respiration involves the oxidation and reduction of electron carriers. Let's start with definitions of oxidation and reduction, and we'll get to electron carriers later in this movie and in parts 2, 3, and 4. In simplest terms, oxidation is the event when a molecule loses an electron. When sodium loses an electron to become positively charged, sodium has been oxidized, oxidation has occurred. Reduction is the event when a molecule gains an electron. When chlorine gains an electron to become negatively charged, chlorine has been reduced, reduction has occurred. Oxidation is the loss of electrons. Oxidation is loss. Reduction is the gain of electrons. Reduction is gain. You may need to study this slide and the next. In this diagram, we can see methane, an organic molecule, being burned, combusted, to produce CO2 and water. In some ways, this is a lot like respiration, except that respiration is a controlled release of energy, whereas the combustion of methane is a very fast release of energy. Methane, an organic molecule, somewhat like glucose, loses electrons to oxygen in the combustion process. This is oxidation. Methane is oxidized. Oxygen gains electrons along with hydrogen ions to become water. Oxygen has been reduced. Now I want you to notice how the position of electrons has changed. Before combustion, the electrons are further away from the nuclei of the atoms. After combustion, the electrons are closer to nuclei. You can see how far the electrons are from nuclei in methane and with respect to oxygen. Yet notice how close to nuclei they are here and here. Oxidation reduction reactions are known as redox reactions. And in redox reactions, electrons lose potential energy as they are pulled toward the electronegative nuclei of atoms, such as oxygen. Oxygen is very electronegative and pulls electrons towards its nucleus, as you can see here. And energy is released in the process. Thus, the combustion of methane as a fuel source involves a change in the position of electrons from being further away from the nuclei of atoms to being closer to the nuclei of atoms. Here is the relevant IB syllabus statement. State that oxidation involves the loss of electrons from an element, whereas reduction involves gain in electrons. And I would like to add that the relocation of electrons releases energy used for ATP synthesis. HL students must keep this in mind as we move forward. Now you might naturally ask, how do electrons get far away from the nuclei of atoms, as we can see in organic molecules such as methane or glucose, as I showed you 2 slides back? And the answer is sunlight. When sunlight is absorbed by chlorophyll, it bumps electrons to positions further from nuclei as the organic molecules of photosynthesis are formed. Once electrons sit further away from nuclei, as they are in large organic molecules, such as glucose or methane, they have high potential energy. Once the organic molecules are broken down, as in cell respiration, the electrons fall back to release energy, the energy that is used to synthesize ATP. Once again, sunlight bumps electrons to higher energy orbitals, sometimes called energy shells, and respiration allows electrons to fall back, releasing the energy required to make ATP. Here is the next IB syllabus statement for all students. Draw the structure of a mitochondrion as seen in electron micrographs. Label the drawing appropriately. You've seen this image already. Cell respiration has an input of an organic molecule, glucose. As well, cell respiration requires an input of oxygen. Carbon dioxide and water are the products to be released as waste from the cell. The mitochondrion has an outer membrane and a second inner membrane that is quite folded. The interior solution is called the matrix. The inner membrane, that's quite folded, is the region where most ATP is synthesized. Here is a diagram of a mitochondrion. You can see the outer membrane. You can see the inner folded membrane. The folds of the inner membrane are known as crista, singular, or cristae, plural. There is a space between the two membranes known as the intermembrane space. It's a solution rich with hydrogen ions. More on that later for HL students. The solution toward the center is called the matrix. Enzymes that synthesize ATP are located on the inner membrane facing the matrix side. Once again, you can see the outer membrane. You can see the inner membrane that's folded. Notice that the intermembrane space is rich with hydrogen ions, and ATPases are located on the inner membrane facing the matrix. Again, here's the micrograph of the mitochondrion. I'd like you to study this image, and I'd like you to study the cell respiration equation. In case you're wondering about water, on both sides of the equation, let me explain. All of the oxygen taken up in cell respiration goes to the formation of water. All of it. All the oxygen goes to water. So if there are 12 atoms of oxygen in these 6 molecules of oxygen on the left, there must be 12 atoms of oxygen in the water on the right. There are 12 atoms of oxygen in these 12 molecules of water. But with 12 molecules of water on the right, the equation is not balanced unless we have 6 molecules of water on the left. Now, this is a simplistic explanation for the presence of water on the left, but for now, it's sufficient. The outer membrane separates the mitochondrion from the rest of the cell. It compartmentalizes aerobic respiration. The matrix is a fluid within the mitochondrion with enzymes for the Krebs cycle and link reaction. These will be understood later by HL students. The matrix has 7ds ribosomes and naked DNA. You should remember the endosymbiont theory. This should be familiar to all students, SL and HL. The intermembrane space is where there's an accumulation of protons, hydrogen ions. There's a proton gradient for a process known as chemiosmosis, again for HL students only. The cristae, the infoldings of the inner membrane, are projections of the inner membrane that increase the surface area available for oxidative phosphorylation, again HL students only. The inner mitochondrial membrane is where the electron transport chain ATP synthases, the enzymes which make ATP, are located, and these carry out oxidative phosphorylation. HL students, you will see these terms again, but to introduce yourself more fully to them, you can spend some time studying this slide. Here is an IB syllabus statement that should be self-evident. State that aerobic respiration requires oxygen and gives a large yield of ATP from glucose. Notice that ATP is not a direct product of cell respiration. As I've mentioned, the energy released from cell respiration is the energy used to drive the synthesis of ATP. The role of oxygen is critical in the production of as much ATP as possible. But there is a type of respiration that does not involve oxygen, known as anaerobic respiration. SL students, you need to bear down on anaerobic respiration along with HL students. Anaerobic respiration will be covered in Part 2. Let me transition towards cell respiration Part 2 by introducing you to the 4 large parts of cell respiration that we need to study, but mostly HL students. Glycolysis starts the breakdown of glucose and occurs in the cytoplasm. The link reaction occurs as molecules enter the mitochondrion, but the link reaction is not shown in the diagram. The citric acid cycle, also known as the Krebs cycle, occurs in the matrix of the mitochondrion. And lastly, the electron transport system occurs on the inner membrane of the mitochondrion. Glycolysis will be the subject of movie Part 2. Glucose is energized with phosphate groups at the expense of ATP. Then the phosphorylated glucose is broken down in a controlled series of reactions that produce some ATP. Molecules of NADH are also formed. NADH is known as a coenzyme, and it serves to carry electrons. This should take you back to an earlier IB syllabus statement. NADH is an electron carrier. Pyruvate or pyruvic acid is the end product of glycolysis. Remember, glycolysis happens in the cytoplasm. Remember, cell respiration is the controlled release of energy from glucose or other organic compounds to form ATP. Cell respiration occurs in stages. This is the controlled release aspect of glucose breakdown: glycolysis, link reaction, Krebs cycle, electron transport system. Here are the 4 stages provided in writing. Part 2 covers glycolysis and the link reaction. Part 3 covers the Krebs cycle. And Part 4 covers electron transport system. Ultimately, 3 of the 4 stages produce ATP: glycolysis, Krebs cycle, and electron transport chain. Carbon dioxide, as well as electron carriers known as NADH and FADH2, are produced. NADH and FADH2 are coenzymes that carry electrons. Their role is to shuttle electrons to the electron transport system, all in the production of ATP. HL students, we will account for all of this in movies parts 2, 3, and 4. And that brings us to the end of IB Bio, Cell Respiration Part 1. Remember, SL students, you need to watch both Parts 1 and 2 to cover all of the material for which you are responsible. You can ignore what's labeled HL, but nothing else.